as evidence of Larry's generosity of spirit, the first speaker he selected is Mathias Uhlen. And Mathias, I'll let you read the biographies. I'm not going to read through them because it would take half of his talk. But I think Mathias, who does work in protein antibodies, is sort of, you know, the antichrist of the gold universe. But nevertheless, gets to lead off. So we're looking forward to it.
Thank you very much. So it's wonderful to be here, to be in Boulder, and also to actually speak after one of my big heroes in science. And Larry has been a hero all the way back to his Aptamir days and now to his Sommarmir days. So it's wonderful to be here. So what I'm going to talk about today is a project which is led from Sweden, and it's to try to characterize the building blocks of human beings, the proteins, and how you can do that in different ways. I'm leading a center, a very new center in Sweden called Science for Life Laboratory. It's an infrastructure for translational research where 3 universities have come together and start to work with technology-driven research in medicine, genomics, proteomics, bioimaging, but also a lot of focus on bioinformatics. And one of the challenges, as I see it, in this new era is to turn a lot of this data into knowledge, and we still have problems with that. So right now we have in 2 years assembled about 400 people, 400 researchers, and about 1/3 of those are in bioinformatics. And just a month ago, our government announced that they're going to put a lot of funding on that, and that was quoted both in Nature and Science. One of the reasons why we are doing this is that we have a lot of biobanks, and just like Larry was talking about, we are very fortunate that we have a lot of good medical records, and then a lot of collections of records, but I will come back to that. And as you can see here, many of the existing biobanks in the world are actually in Scandinavia. So what we try to do in this new center then is to work with that biobanks, trying to teach how we should treat patients and so on, and I will come back to that very soon. So what I'm going to do then is to give you an introduction to what I call personalized medicine. I'll tell you a little bit about the Protein Atlas project, and then go into a little bit about technology and give you a reason why we are doing this, and then come back in the end into more clinical biomarker discovery. So first, a little bit about omics. I think it's very fortunate to be a scientist in life science right now. This is my personal kind of view of what has happened in the centuries where mankind has tried to systematically go through nature. And that was astronomy in the early days, biology. Of course, the 19th century was the chemistry. I think the last century was very much the building blocks of the material, the physics. But right now, we are in this incredible age where we will go through all the building blocks of human life, but also other types of life. And as you all know, the building blocks of life is the DNAs and the proteins. And now we have tools to study that in an incredibly pace, and it's called omics, just like Larry was saying. So we try to use that to sort of get to the parts list of human life. This is a little bit of a self-boosting slide, because one of the technologies that was preceding this new generation of DNA analysis is the pyrosequencing that we published in Science in '98, and that then led to the 454 instrument, and then to a lot of other instruments. And the pace now and how cheap it is now to do a human genome is just incredible. And this has led then to this way that you can actually start thinking about sequencing everyone in this room, and maybe do that in more or less in a couple of weeks. And we can all sit in front of our computer and look through our genome and see what is wrong with us. And that's kind of scary, maybe for some. And I'm very proud then that Jim Watson, the person that was first sequenced, was done using the technology coming from our lab. So what have we learned then from genomics so far? Well, and these are just some of the stuff that we are sequencing in our lab, and it's kind of funny that the Christmas tree, the spruce, actually has a genome which is 7 times larger than humans. So try to explain that. It might be more intelligent than you think. But anyway, so what we learn from genomics is, of course, how many genes all of us have. All of us in this room has 20,000 genes that codes for proteins. We know the sequence of the genes and the proteins. We know now very much the variability between 2 people in this room, or between people in the world. And we know the differences between species. And we learned that we're very, very, very similar to monkeys and so on. But we also learned a lot about gene regulation. And that's, of course, very, very interesting. But we have not learned a lot of things from genomics so far still. And the most important, and I think quite astonishing, is that we learn very little about human biology. It's very hard from the sequence to know almost anything about the phenotype, how we are actually working. The functional proteins, we can get some ideas. The signal pathways we don't really are helped with. And especially it has not led to new drug targets. Well, not in a major way anyway. And not even into new genetic markers that have made it into the clinic. And why is that then? And it's a little bit like this guy here standing in front of this haystack. It's very hard to find a needle in the DNA material that you can use. So one could then ask the question, why do we get sick? And I usually ask my students about that, and the most obvious one is, of course, what everyone talks about, and that is genetics. We get sick because our fathers or mothers gave us some hereditary traits, and the second one is also obvious. That is environment. Of course, the genetics and environment combined gives us who we are, but also how we behave and also how we get diseases. But there are 2 more things which are even more important, genetics and environment. Does anyone can say that? One? What? Aging. Behavior. Behavior, I think, is kind of part of genetics and environment, in a way. But that's so one is microenvironment, I think, is part of the environment. So aging, I think, is very important. I think we are very different when we are 5 years old and when we're 85, and we also get different diseases. But the 4th one is even more important. And what is that? Epigenetics? No, I don't think so. I think this is my point, it's stochastic events, randomness. And I never hear any scientists almost talk about that. But if you think about cancer, if you think about infectious diseases, a lot of the even cardiovascular diseases, maybe not autoimmune diseases, but maybe also that, a lot of the reasons for that is probably stochastic events. And I don't think that we think so much about that, but I think some of us do. And this, of course, means that studying the genetics is important, but it's only a small part of the scenario, and you can hear more other things going on. Stochastic event, if we take one thing, for example, cancer, we are getting mutations in our body every second from the oxygen that is here, and sometimes that, if you're unlucky, that gives you cancer. We're passing someone in the street that happens to have a flu, and then he or she passes that over. That is what I call stochastic events. Bad luck. Yeah?
But we think of stochastic events like a butterfly effect, something that gets multiplied and magnified and
although the butterfly effect is really, in a way, things leads to another. In this, I'm kind of sloppy here, just saying sometimes we have bad luck, and it doesn't it's not because we are smoking, it's not because we have genetic defects, and it's not because it's the aging, it's something else, and that's stochastic events. And what I'm trying to say with this is that means that genomics can tell us things, but in order to actually understand what's going on, we need to move to the phenotype, and the phenotype is several things, but one is the proteins, and the other things are the metabolites in the body. And that's why I think from a disease point of view, we of course have to go for the proteins, which are the chemistry of life. And it's a cause of all these things. Proteins are of course also important because 98 at least of the drugs today are towards proteins. We know from the Genome Project that we have 20,000 proteins. We know that all the pills, the drugs in the pharmacy are directed to about 2% of those, and 30% of these proteins has not been studied at all. So obviously we need a way to systematically analyze the proteins. We actually owe this to the scientific community. So the challenge of life science, as I see it, is to take the genomics and move into the proteins and the metabolites and study those and give the society different types of products. Another way that we have been talking about is personalized medicine. The way I see that is that we have to move away from the traditional way of making pharmaceutical drugs, which is an average drug to an average patient, into an individualized drug treatment where we use all these new technologies to stratify patients, group patients into responders and non-responders, and get a little bit more individual treatments. There was an interesting quote in one of the journals last month in Nature Biotechnology that says, and I think some of you have heard this quote before, that an estimated 90% of the drugs currently on the market today only works in 40% of individuals. And this translates annually, and that this is only in the United States, into $300 billion worth of ineffective prescriptions. So I think this is a very good argument that we have to stratify patients in order to get better treatments. And this is the cost of the pharmaceuticals. Then you can also think about all the cost of side effects, which makes this incredibly important for healthcare. So how large is then the human proteome anyway? We can define this in different ways because we have genes coding for proteins, and those are 20,000, we know that. But then the proteins are expressed in the cells, and they get adapted, and they are isoforms and so on. So one can argue about how many they are, but I'm not going to do that. But what we have, some of the people in the society or in the community then has said, this is probably the good time for us now to get together the parts list of human beings. Try to characterize all the molecular isoforms, where they are in the cells and where they are in the tissues, to really try to get to a sort of an atlas of all the human proteins. And how should we do that? Well, some of you maybe have been driving in Paris, and if you've done that, you know it's not very easy. And I think this is a little bit how the proteomics community has debated how to do this, sort of like this is illustrated. But our way of doing this is to use antibodies, very much influenced by these protein binders that Larry has been the sort of king of. And we started a project in about 10 years ago in Sweden. It's very nice to have rich families. So there was a family called the Wallenberg family that gave us then about $150 million to do this project. And with that, then we could start this incredibly project to go through all the proteins that you have in your cells and bodies. And I'm not going to be too technical here, simply to say that this is a technical tour de force where we sending proteins to China and Korea, get it back and purify them. And then we do a lot of analysis, And the way that we have done this is that we put it onto a database, a portal that can serve the whole community, and it's free of charge without any restrictions. With that funding, then, we have assembled a team of 4 sites in Sweden. We have about 20 formal sites in Europe. We have some very important sites in Asia, which we are very proud of. And then we have quite a lot of funding also from NIH and NCI. So we have collaborations here in the States too. The Indian site I'm very proud of. Here we have about 15 pathologists sitting in a room close to the central station in Mumbai. And I don't know if you've been to Mumbai, but it's a pretty interesting place. And in that room, we have Macintosh computers. And you can, of course, send all the images down by internet. So instead of sitting in front of the microscope, they sit through and go through all the images, send them back to us, and then they get a check of $75 or something like that. And this is working extremely well, and I'm very proud of this. So what are we trying to do then? Well, we're basically saying, all of you in this room has 20,000 proteins. We have to know, and we should do that in 5 or 10 years, we should know where every one of those proteins are in the organs, in the tissues, in the cells, and also inside the cells. We call that in the organelles. So we have been working on this now. We had the first release in 2005, and now we have 12 million images which are publicly available. We have about 20,000 visitors every month to this site, and hopefully this is leading to better science in the future. The version 9 of the Protein Atlas was launched about 6 months ago. It had 12 million images, and in a few months, in September, we will release the version 10, which will have 14,000, and we're sort of moving up. And what we said to our funders, the Wallenberg Foundation, is that with the funding we have today, we want to go through another 10 genes and proteins every day, because that means that in October 2015, we will have a first draft of all the human proteins where they are. There's also a lot of commercial companies out there that are selling antibodies. And they have been, all of the big companies have actually sent us their antibodies, and we can then put them into our pipeline, validate those, and see how they're working. And so far, we have got actually 16,000 antibodies from 40 different companies. And these are the success rates you can see here for these companies. So there are some to the left here, which almost 90% of the antibody works. But you also have other companies, and I'm not going to say from what countries they are, that about 10% of the antibodies that they sell to researchers actually works in our hands anyway. And actually, right now, the success rate of the commercial antibodies which are sold to researchers around the world, for us, is about 1/3 of them works. So this is a huge inefficiency again, and therefore we have just launched together with a newspaper in England, Nature, a portal, Antibodypedia, where we try to have a free way for scientists to exchange data about antibodies. I was going to talk a little bit about specificity of antibodies, but I think this is a little bit technical, so I'm going to kind of move here and then go to the next topic and just say, how are we using these antibodies then? Well, one of the obvious ways of looking at that is, where are the proteins in the cell, the subcellular atlas? So what we're trying to do is to say which proteins are in the nucleus, in the middle of a cell, how many are in the mitochondria, the energy factory, and so on. Because if we understand where a protein is, we get a feeling for the function of that protein. But we can also say what proteins are this protein interacting with, and so on. So one of the nice things about having the genome data is that we can look at that and do bioinformatics. And we can predict for example, how many proteins are in the membrane of the cell. And we published a paper a few years ago where we say that about a quarter of the proteins in our cells are sitting in the membranes. And those are very important because they are the targets for almost all drugs. Then recently we also looked at how many proteins are secreted out from the cells. These are the proteins which are swimming around in our blood and are also signaling when we are getting infections and so on. And without going into the details, I can just say that we have about 16% of our genes are actually secreted, and actually 40% of the proteins in our bodies are either going out or are sitting on the surface of the proteins. But also what is rather remarkable, almost 20% of the genes in our bodies, they have proteins that goes to different locations in the cell. And I will tell you that we almost know nothing about why they are doing this. In some cases, IgG and so on, we know, but in most cases, we have no idea why proteins are going to different locations in the cell. But we also have this fantastic resource, if I'm allowed to say that, of antibodies, so we can actually see the proteins in our microscopes. So this is just an image of an unknown protein. No one has seen it before. We paint it in yellow or green, so you can see that this protein is sort of involved with the actin filaments, the skeleton of the cell. Here you have another protein. You can see a totally different type of structure. It's a protein that we think is in the peroxisomes. There are also other proteins which are in the nucleus, and this is a beautiful picture of an unknown protein which are sitting in a very, very interesting way in the nucleus of the cell. The nucleus is blue here. And then you have other proteins which are involved in getting the chromosomes right and so on. And you can see this single dot, yellow dot there in the middle, which is a protein, an unknown protein, which probably is involved in the centrosomes. So we can do this very detailed. There are new technologies, the knockdowns with siRNA, which got the Nobel Prize. We can also knock down the staining and really get very good quality. We can also complement this with GFP fusions. We can clean up the literature, because here we have, for example, a protein that the literature says it should be in the mitochondria, but both with our antibodies and with this GFP, we know now that it's in the cytoplasm. So what we hope to do in the end is to create a subcellular atlas that shows where are all the proteins in the different parts of the cell. And here you can see this kind of complicated plot. Every dot here, colored dot here, is a protein. If it's blue, it means that this protein is only in one location. If it's in green, it's a protein which is at least in two, it is in two locations. And in the end, then, we can have these subproteomes, all the mitochondria proteins, and this hopefully will be great help for all of us that tries to understand biology. So what we try to create then in this Swedish project, which is also funded a little bit from the NIH, is to have a first draft of the Subcellular Atlas in 2015, and then to try to move for another 5 years to clean this up, to have a better, more quality assured atlas of all proteins. And one of the nice things about doing this with all the data in the public domain is one could come together in different international collaborations to really try to get this as a starting point for then future research. Then we also work with tissues. And it's of course very interesting to study where are proteins, which proteins are in the brain, which are in the kidney, and so on. And here we used a wonderful technology that was developed at Bethesda a few years ago called tissue microarrays that allows us to take material from different parts of the body, put them together on a single glass slide, paint that with our antibody, and then get very pretty pictures that tells us where are they in the kidney, where are they in the brain, and do this in a very, very systematic manner. So you get very pretty pictures like this one. This happens to be one of my favorite proteins, CD47. And you can see then in brown the cells that have this protein on its surface. I think this is the fallopian tube of a female aged 23. And one of the astonishing things that we see very often is that when we are looking with this very high resolution where we can see single cells in the body, what you can see here is that some cells have this protein, then there is some neighboring cells that looks identical, doesn't have it, and so on, and we still don't know what this is really about. And here's some other examples of all of these proteins here are unknown proteins which has a very specific expression in the body, and they have this microheterogeneity which we find very, very interesting. So all of you can this afternoon go home, Google Protein Atlas, and then go in and look for your favorite proteins. And I urge you to do that. And then you can click to the summary page, and you will get information about where the subcellular localizations, where they are in normal tissues, and so on. And the beauty of using the web is, of course, that we can actually put all the primary data, the images, there. So that's why you can click and actually go and see, this is how our antibody looked, and this is the annotation the Indian pathologist gave you, and then hopefully you can find some wrong things and you can send me feedback. But this is, of course, what we want to do. One of the driving forces for this is to try to find proteins which are very tissue-specific. What I really thought when I started this project 10 years ago was that we can now define the proteins which are in the kidney and gives us a filtration apparatus, and what are the proteins in the brain that can give us the memory, and what are the proteins in the liver that gives us, you know, the detoxification factory and so on. So I'm not sure we were disappointed. I guess it shows that you can never know what biology takes you. But we published a paper about 2 years ago in our first analysis, and where we saw that there were extremely few proteins which are tissue-specific or cell-specific. As a matter of fact, you can of course not read that list of 84 proteins, but they have all the famous ones, the troponins and the insulins and the PSA and so on, but there are very few others. The nice thing about it, that there were quite a few new ones. And together with people here in the States, we have, for example, looked at this protein, CIR1, that seems to be extremely specific for the heart muscle, and that actually has started a lot of collaborations trying to understand what it's doing in the heart. We've also been trying to move into other technologies, and one is, of course, mass spectrometry that we do with Matthias Mann in Munich. But also this wonderful new technology of transcriptomics, which is using this DNA sequencing to look at the RNA in the cells, because the genes is transcribed to the RNA, and then the RNA is translated into the proteins. So looking at the RNA is a fantastic way of looking at what is then translated into the proteins. And now, of course, you can also take these same samples and also do genomics and sequence the genome. And again, I'm not going to be too technical here, but we find very good correlation between RNA and protein changes, which is nice. We also find something rather peculiar. We call it the inside-outside rule, and we published this again about 2 years ago, where we saw that a lot of the proteins are expressed everywhere in similar, and those are very often intracellular proteins inside the cell. But then when you looked specifically at the proteins which are changing between different tissues, they're almost always, not always, but very much more proteins which are on the outside of the proteins. So it kind of looks from a superficial point of view, or that the outside of the cell is what is different, while the inside is sort of household functions. There are, of course, a lot of exceptions to the rule. But by having all this data, you can do this, what is called systems biology, this kind of bioinformatics. So you can compare the expression, the number of of building blocks in different tissues. And to the left here, it's a comparison of, I think, about 14,000 genes and proteins in the liver and in the tonsil, which are very different organs, and how they relate to each other. And I don't have a pointer, but what you can see then, I don't have time to explain it, is that almost all of the proteins are expressed in both tissues, and there is a small amount of proteins which are tissue-specific for the liver and the tonsil. And this has allowed us then to actually now start classifying all the genes. So all the 20,000 genes now, we can say these are household proteins, or at least expressed in all tissues. It's about half of the genes, and then start to define the tissue-specific proteins. And then you can move, since we have the tools to look at the proteins, we can then go in and look and see these kind of pretty pictures of proteins in the liver and the tonsil, and I'm not going to go into the details of that. But the lesson we learn from this is, to me, a very big surprise. And what we seem to see is that a kidney is not a kidney because it has a bunch of household proteins and then it has kidney-specific proteins that forms a filtration apparatus. A kidney has the same proteins, more or less, as the brain, but it's in different concentration from the brain. And it is the way, the local concentration of those proteins that then forms probably complexes that are specific for the kidney. So we are not more than kind of Lego pieces, where we have the same Lego pieces in almost all cells of the body, and then, of course, there are some which are specific. So to me, I'm a little bit more puzzled about human biology now than I was 5 years ago, but it's, of course, very exciting that we can start to assemble this parts list. So again, we hope to have a tissue atlas then with the first draft in 2015, and then we hope to clean this up to have a more quality short atlas 5 years later. It's also important that we're creating a reagent toolbox for scientists, and we have collaboration with the company in St. Louis, Sigma Aldrich. They are selling these antibodies, and there is about 100 of these antibodies going out every day now to scientists. And there is almost a publication every day of the use of these antibodies in human biology. So I'm going to end by just saying a few words about biomarker discovery, because we do have then, we can now start moving into a more systematic analysis of biomarkers. And I can say a lot about biomarkers, that you have diagnostic markers, prognostic markers, and predictive markers. We have sort of focused on cancers because it's very easy for us to get the material, and we're going into all these different types of cancers. In the protein atlas, you can do very clever biomarker discovery yourself, and you can all do it at home tonight. For example, if you ask, how many transcription factors do you have on chromosome 14, which are only expressed in, or which are expressed in the skin? I'm sure you're interested in that question. Then you will get, I can't see how many hits, but about 6 hits. And then you can actually go in and look in these images and see the transcription factors are in the nucleus and they are in the skin. Maybe a little bit more interesting is then to ask, show me proteins that could be potential biomarkers for predictive medicine. And here's just an example where we have asked, show me all the proteins where some breast cancer patients have a lot of it and some has none of this protein, because that means that we can group the patients, stratify them, and then we can compare that with their outcome. And it's nice when you do that in sort of in silico search, that one of the ones that are popping up here is the estrogen receptor, which is a known marker like that. So we have done this, and we now have quite a few of these biomarkers. I just want to show you one, RBM3, which we are very, very excited about, which is it's a protein that correlates very well with the efficacy of adjuvant therapy, not only in breast cancer, but also in some other cancers like ovarian cancer and colon cancer. And finally, I just want to say a little bit about biomarker discovery in blood. This is I think the most exciting way of diagnosing people. It's very easy to get blood. It's a non-invasive way of doing it, and we have a lot of, we are all giving blood in different ways. The problem we've had with the biomarker discovery of blood is that we've been using a technology called shotgun MS. And the sensitivity of this method is a little bit symbolized by the iceberg to the right on this picture, that we can see what is sticking up, which are the most high abundant proteins, but we cannot see what is under the sea level, which is the lower abundant proteins. So now a lot of people are trying to get more sensitive assays, and we have a very good company here in Boulder who's doing this very, very well. So with that, we have started to collect from different clinical doctors a lot of disease cohorts, and I just love this image because it shows us how our biobanks are looking when they come from these proud doctors in these different diseases. And if you can see some sort of standardization here, you must be very clever, because I can't. So one of the things that we really desperately need is standardizations moving into this, and this is happening both in Europe and here in the States. So now there are a new wave of biobanks being formed where you have a standardized way of doing this. And with that, you can start doing a lot of different things. And I thought I would just show you one example. We have been interested in toxicity markers, because actually toxicity remains the chief cause for failure in the clinical development trials. Together with AstraZeneca, we have looked at kidney toxicity and been looking for proteins that leak out from the kidney when you're starting to get toxicity. And without going into the details, this is just one example, a protein called fibrillin-1 that we are which we are now going into clinical trials with. So with that, I want to then conclude by saying that this is an absolutely amazingly part of history of life science. This is the decade where all the building blocks of human beings will be characterized and the parts list will be assembled. I have a feeling that this will be a lot of data, and I have a feeling that knowledge might come along, but it might take a long time before we actually understand what's going on between these magical moments when the proteins are meeting in the middle of a cell and something happens. But anyway, we will release the first draft of the human protein by 2015. We are creating a comprehensive toolbox that everyone can use, but I think what even more exciting is that we're defining the building blocks of human beings. And then I want to thank all the people that have been involved in this. This is the Swedish team that you see here, but there are also other people then in different parts of the world, and we're very grateful. And then, of course, as always, you cannot do anything without funding, and therefore I'm very grateful to the Wallenberg family for giving us this very generous funding. Thank you very much for your attention.